Get to look a little at the physiologic basis. Let's look at some molecules moving around. Okay, first off, water. Easy. Water moves entirely by diffusion. As I've told you before, there are no water transporters, right? The closest thing we have are aquaporins, but they're just dumb doors, right? We don't have a pump for water. So water is going to follow the osmolarity, okay? And osmolarity plays a big role in digestion. If you imagine we're going to make up a story. You've got a beaker and it has 3 molecules of protein in it, right? Just 3. So its osmolarity is very, very, very low, right? Now, we add protein digestion enzymes, and our 3 proteins become 100 million amino acids. How has the tonicity changed? Yeah, we went from very low tonicity to very high tonicity just by digesting something, right? Because we took 1 particle and made it 100 million. See? So osmolarity changes dramatically as we go from stomach to anus. Now, thankfully, we don't have to think much about that because the water is just going to move accordingly, right? So like early in digestion, before we have a lot of particles, the osmolarity is quite low, so we end up absorbing water, right? Later on in digestion, when the big polymers have become monomers, the water moves in the other direction, right? Towards the area of higher tonicity. So we get water moving in and out, and it just follows its track. Now, because early in digestion, osmolarity in the lumen tends to be quite low, it means when you drink water, it's absorbed very quickly, right? Because the water is absorbed early before the rest of the materials have had a chance to fall apart or get digested and increase the tonicity. Short version, water is absorbed very quickly. Like if you drink water, it's in your blood very quick, all right? The only exception to that would be if you also took in a very hyperosmolar material, which we don't like as humans. Like when the osmolarity is too high, we think it's too sweet, too salty, whatever, we'll push it away, right? Like corn syrup, right? Corn syrup has a very, very high osmolarity. Would you eat it? Maybe not directly, right? You dilute it down first. It's the whole idea. I know you didn't know that you thought in osmolarity, but you do. At least part of your brain does. Okay, so water moves by diffusion. Ions, we're always talking about ions in this class, right? Ions are moved by active transport, co-transport, or diffusion. So there's a mixture. Yes, we have the sodium-potassium pump doing its usual thing. Now, importantly, for the digestive tract, the sodium-potassium pump is keeping the sodium concentration in these cells low, which sets us up for co-transport, like we saw in the kidney, right? Same idea, where sodium falling down its concentration gradient can move amino acids up its concentration gradient. Same here, sodium moving down moves glucose up. Sodium transport is fueled, it's an active process. Chloride is passive because it's following the electrical effect, right? You know, remember, chloride is really quite neutral in the body. It just moves according to the charge. Co-transport of sodium sodium-potassium pumps fuel the co-transport of those other materials. We got to talk about hydrogen ion and bicarb. This is a rare example where we see both going in the same direction, right? We've seen these move a lot, but usually they go in different directions. Here's an example where they're going in the same direction. Okay, so bicarb is being pumped out in exchange for chloride. Hydrogen ion is being pumped out in exchange for sodium. Now, what would happen when those two things mix? Well, they're going to neutralize each other, right? We're going to get water and CO2 out of that. And then the CO2 oftentimes is scooped back up by absorption, taken to the lungs and exhaled, right? Because remember, the GI tract has a very big surface area and plenty of blood flow, so any CO2 in there is going to get whisked to the lungs pretty rapidly. Aldosterone, I know you're like, oh man, still aldosterone. Yes. Aldosterone promotes sodium absorption. And it does so by installing additional sodium-potassium pumps, aldosterone-sensitive sodium-potassium pumps on the basolateral membrane, right? So more pumps means more pull in, right? This makes sense because aldosterone, of course, is our sodium retention hormone, right? So if we're in a situation where we're holding on to salt to hold on to fluid, well, we want to pull the salt we can out of the GI tract, right? One of the strange things in our human existence is that for us, sodium is like in plentiful supply, right? That isn't true for all the other animals. Like minerals are a big deal. Anybody heard of a deer lick or a cow lick, right? It's because the animals will seek out salt and lick on it because they don't have enough in their diet. Where we as humans have so much of it, it causes us health problems, right? And that's because, of course, we haul it up from the ground. We mine it out, and the cows don't have, dynamite. Ion absorption. Okay, so the bicarb is exchanged for chloride. We just looked at that. Come back here, right? Bicarb is exchanged for chloride. Sodium is exchanged for hydrogen ion. Why put bicarb in the gut? A few reasons. In the large intestine, bicarb secretion is meant to neutralize acids produced by bacterial metabolism. I think we chatted about that the other day. In the rest of the body, or in the rest of the intestine, the bicarbonate and the hydrogen ion are going to vary according to what's being eaten and what the pH of it is and all that kind of stuff. Short version is this bicarbonate and hydrogen ion secretion helps us to maintain the pH of the gut where it needs to be, right? Because if you have both hydrogen ion and bicarbonate, well, if the pH is too high, well, you have hydrogen ions that are going to dilute that, and if it's too low, you have bicarbonate that's going to neutralize that.